Cancer is one of the world's deadliest diseases. While treatments like chemotherapy, radiation, and immunotherapy have saved lives, they still face major challenges: treatment resistance, immune suppression, and harmful side effects. CRISPR-Cas9 changes the game. This precise gene editing tool can upgrade therapies like CAR T cells, antibody drug conjugates, and tumor vaccines, making them more targeted, safer, and more effective. Today, we'll explore how CRISPR-Cas9 is transforming cancer immunotherapy and paving the way for better treatment outcomes. Genome editing is transforming molecular biology, and at the forefront is CRISPR-Cas9, which is short for Clustered Regularly Interspaced Palindromic Repeats, a precise, adaptable, and cost-effective technology that has surpassed other tools like zinc finger nucleases and talents, as verified by Al-Jabali 2024. It was originally discovered in bacteria as an immune defense. CRISPR-Cas9 is now used in medical research, gene therapy, and even crop improvement. The CRISPR-Cas9 system has two key parts: the Cas9 protein, which acts as a molecular scissor, and the guide RNA that directs Cas9 to a specific DNA sequence. Once there, the Cas9 makes a cut and the cell's repair machinery modifies the DNA through processes like non-homologous end joining or homology-directed repair. Its simplicity and accuracy make it invaluable, but challenges remain, such as off-target effects, delivery into cells, and ethical concerns, especially in human germline editing. Now that we know how CRISPR works, let's see why it's a game changer from its development. The CRISPR story begins in 1987, when scientists first noticed strange repeating DNA sequence in E. coli. In the 2000s, they discovered the sequences were part of a bacterial immune system storing fragments of viral DNA to defend against future attacks. So before CRISPR, gene editing relied on earlier tools such as zinc finger nucleases, which developed in 1996, which were precise but expensive and complex, and talents, introduced in 2010, which were effective but labor-intensive. Then in 2012, Jennifer Doudna and Emmanuel Charpentier demonstrated that the Cas9 protein could be programmed to cut almost any DNA sequence. So CRISPR-Cas9 was fast, affordable, and easy to program, marking the start of a new era in gene editing. Today, several powerful immunotherapies are being used to fight cancer. First, cancer vaccines work by training the immune system to recognize and attack tumor-specific antigens. Second, oncolytic viruses are engineered to selectively infect and destroy cancer cells, while also triggering a broader immune response. Third, immune checkpoint inhibitors removes the brakes on immune cells, allowing them to mount a stronger attack against tumors. Fourth, CAR T cell therapy involves reprogramming a patient's own T cells to seek out and destroy cancer cells more precisely. Finally, antibody drug conjugates use targeted antibodies to deliver potent chemotherapy directly to tumor cells, minimizing damage to healthy tissues.
Based on Brokowski and Adley's 2018 review on CRISPR ethics, applying CRISPR-Cas9 in cancer immunotherapy demands careful attention to key ethical considerations. First, there are safety risks, off-target effects, mosaicosm, and unpredictable long-term impacts. Second, we must distinguish between somatic editing, which is generally accepted for patient treatment, and germline editing, which is heritable and raises consent and multi-generational impact concerns. Third, equity and access are critical, as high costs could widen healthcare disparities. Fourth, there is a risk of misuse, including non-therapeutic enhancements and a potential revival of eugenic practices. Finally, the authors stress the need for a strong regulatory oversight, informed consent, and continuous risk-benefit reassessment, and global ethic frameworks to ensure CRISPR use is both safe and socially responsible. In this groundbreaking phase 1 trial, scientists used CRISPR-Cas9 to engineer patients' own T cells to better recognize and attack cancer. They deleted two genes, TRAC and TRBC, to prevent interference with a cancer-specific receptor, and removed PDCD1 to overcome immune exhaustion. They then added a synthetic TCR targeting the tumor antigen NYESO1. Three patients with advanced, treatment-resistant cancers received these cells after chemotherapy. The results were very promising. Engineered T cells resisted in the body up to 9 months, reached tumor sites, and caused no severe side effects. One patient's tumor shrank significantly, and the others had stable disease. This just shows that CRISPR-edited T cells can be safe, durable, and biologically active, paving the way for more powerful cancer immunotherapies.
So imagine having scissors that can cut DNA precisely. That's what CRISPR is. Now let me tell you how it works. So CRISPR takes inspiration from the natural defense system of bacteria. So when viruses infect a bacteria, a portion of their viral DNA gets stored as part of the bacteria's own DNA called the CRISPR region. So this will serve as a genetic memory of the virus, which will allow the bacteria to recognize and fight it off in the case it attacks again. So the researchers, they took advantage of this bacterial defense system. They created guide RNA or gRNA that complements a specific nucleotide sequence in the bacterial DNA. So through this, the scientists will be able to fix or kill a gene. Here, the gRNA pairs with a protein called Cas9. So this Cas9 would be our DNA scissors. The gRNA would serve as a guide for Cas9, directing it to the exact region in the DNA whose sequence it matches with. So the gRNA Cas9 complex, this then finds the target DNA sequence through a DNA signal called the PAM, or protospacer adjacent motif. So Cas9, your DNA scissors, then precisely snips both strands of the DNA just right where the gRNA is bound. So when the cell detects a break in the DNA, it will employ the DNA repair systems, of which the researchers can exploit for gene editing. There's the non-homologous end joining, wherein the cells will join the broken ends of the DNA. So this has risks for mistakes, which can unintentionally knock out the target gene. And there's also homology-directed repair, where if the researchers provide a template of DNA with the desired sequence, this can be used to fix the break precisely. So this allows for new genes to be inserted. So in order for this gene editing to take place, the gRNA-Cas9 complex must first find its way into the cell. So researchers have utilized several methods to do this. Let me show you some of them. First, we have viral vectors. So these are engineered viruses such as AAV or adeno-associated virus. We also have lentivirus. So these can be used as delivery vehicles because they can carry the DNA instructions for CRISPR in the cells. They're efficient because they're naturally good at penetrating cells and they have specificity when targeting tissues. So lipid nanoparticles, these are tiny fat bubbles that can also be used to introduce the gRNA-Cas9 complex into the cell because they easily fuse with cell membranes given their lipid nature. We also have electroporation. Electroporation is also used by researchers, and this is done by applying electrical pulses in short intervals. These temporarily creates openings in the membrane of the cell. Despite its efficiency, however, electroporation can damage the integrity of the cell. Next, we have microinjection. So this is a direct form of delivery where fine needles are used to inject the components of CRISPR into the cells. So this is mainly used for single cells and embryos. So these are just some of the delivery methods for CRISPR into the cells. There are still emerging delivery methods such as cell-penetrating peptides, nanoparticles of polymer, and even mechanical forces. Despite CRISPR's advancement, it still has its own challenges and limitations. First, off-target. So since CRISPR cuts DNA, it has the tendencies to cut at places other than its target, which can lead to unwanted mutations or even harmful changes. Another challenge for CRISPR is its delivery method. So delivery method should be safe as much as it's efficient. It should also be able to target tissues precisely. So next, we also have double-strand breaks and risks for safety. So CRISPR works by cutting both strands of the DNA. So sometimes this could lead to unintended apoptosis, chromosomal damage, and even cancer. So next, immune responses. So one of the challenges in CRISPR is the immune response it can elicit. The immune system might recognize it as a foreign body and attack it. So this could heighten the risk for gene therapies. Lastly, CRISPR faces ethical considerations, especially when it's used in human embryos. It raises ethical questions regarding the misuse of it and the consequences it comes with.
What are the future directions or future prospects of CRISPR-based cancer immunotherapy? Well, there are two possible directions, which are the incorporation of spatial genomics and nanotechnology. So let's dive into each and discuss more about them. So first off is spatial genomics. In the context of tumor growth, cancer cells continue to proliferate with the aid of a surrounding environment called the immunosuppressive tumor microenvironment, or TME for short. The TME is composed of immunosuppressive cells and molecules that inhibit immune cells from attacking the tumor. With such cases, spatial genomics is a separate approach that investigates how various genes work and how they interact with one another within the physical location or tissue that they are in. This approach is potentially seen to enhance the CRISPR-based cancer immunotherapy, as this will allow scientists to observe complex interactions within the TME. Now, let's move forward to the next one, which is nanotechnology. So with specificity and accuracy being among the challenges of CRISPR technology, the use of nanotechnology has been sought to potentially address this limitation. So nanotechnology or nanobiotechnology focuses on the study and utilization of matter or particles at the nanoscale for biological and biomedical applications. So in applying this to CRISPR technology, tiny engineered particles called nanocarriers can cross cell membranes and deliver the CRISPR components into the cells more effectively. So compared to viruses, which are generally used for delivery, the nanocarriers are generally safer and less likely to cause harmful mutations. So one type or one example of this is the magnetic nanocarrier, which can be steered or to specific locations in the body using external magnets. This allows scientists to control exactly where CRISPR edits happen. So now let's talk about the possible impact of CRISPR-based cancer immunotherapy on healthcare. So for cancer in the Philippine context, it is considered as the second leading cause of death in 2024, accounting for around 38,132 deaths, which is 11.1% of the total mortality during that year. And so that shows how cancer is very prevalent in our country. And so a lot of experts find it very crucial to find an efficient and effective treatment for such a detrimental disease. And so enhancing treatments such as the immunotherapy with the aid of CRISPR-Cas9 technology can heavily impact the state of healthcare in the country. First, it will revolutionize cancer immunotherapy with the following attributes. So first, it would be more efficient since CRISPR would allow for precise editing. And then secondly, it would be more personalized and CRISPR allows to identify and target new genes and pathways involved in cancer development, leading to the development of novel cancer therapies. And then lastly, it will lead to reduced side effects since there's less damage to cancer cells. And all of these would result to better patient outcomes. However, the successful implementation of the Philippine healthcare system requires addressing challenges related to accessibility, affordability, and ethical issues. These barriers include financial constraints for patients, along with the need to balance quality care and cost control, and ensuring fair and transparent healthcare practices.